so far? Great. So far, so good. That's a good answer. We'll start 10:05 since you had a session before me. I'll give you a little time. So 2 more minutes, everyone. One more minute, everyone. Then we start. Okay, guys, let's get ready. So you can hear me, right? And you can see the PowerPoint, everyone. Let's get in our Zen. Everyone can see the PowerPoint and you can hear me well.
Yes.
Awesome. So it's 10:05. Please see what's my plan today? My plan today is to review regulation of gene expression 1 and 2, fibrous and glycoprotein. Please, I'm not teaching you today, so I'm going to just bring back your attention of what you have learned so far. And then I would like to wrap this up in an hour. And the next hour, I will open for question and answers. I want you to do well. I have given you help. Please take help, and I'm sure you're all going to do well too. So far, you have been doing good, but let's see how we can combine hard work and smart work with my help and help you get good grades. Right? So please, first of all, you should be aware of your lectures. You have 4 lectures and 2 CVs. Every lecture will be tested. There are 16 questions. There will be 4 questions per lecture. One of the CV will be tested, as you always know. I like first bringing your attention to what you need to do. Yes, there are some quizzes which are combined and they will be reopened in CP module, which is also an advantage for you to redo that quiz again and increase your grades. So don't let those quiz topics confuse you. These are your 4 lectures for MSK2 exam, and these are the 2 CVs. Please see, I have offered help. So let's see what your are help available if you want to grab them for last minute review. I have been given the recaps for regulation, fibrous as well as glycoprotein to your class reps. That is in the MSK2 help folder on the drive. Line diagrams for last minute review, if somebody wants to do, is also on help folder on the drive. Your MSK2 topic list, disease list, and your review PowerPoint are on Canvas. in the respective MSK2 module. Your weekly interactive sessions are posted. Also, they are a point of focus if you want to do those questions. Your self online assessments are open for learning and your practice quiz. Please, please go from focus to outside now. All the focus is available. You may choose any focus to get good grades. Please take help. Let me know if you're missing any of these files and they are not available. I will make them available. But stick to your learning objectives. They are the best guide to your test. Learning objectives are tied to your test. Now, what was the focus of regulation of gene expression? I do have my pointers. They can be just reviewed before the exam. Okay, did I do the motives? Oh, did I do the transcription factors? What are cis elements? So they are just pointers. They are tied to your learning objectives. Either you can do learning objectives or these pointers. So what was the focus of regulation of gene expression? I'm not teaching you. I'm bringing you back to the focus. I have a question, couple of questions also along with the lectures. There is no quiz due this Sunday, right? I don't think so. I will check. I will check, please. I will check the quiz. Let's first do the review. What was the focus of regulation of gene expression? First of all, gene regulation is controlled at the level of initiation transcription. Secondly, gene expression is controlled at the level of co and post transcriptional processing. Thirdly, regulation of gene expression is controlled at epigenetic modification. And lastly, it's RNA editing. These are only the four focus points around which you have to beat. You have to beat around them, but bring every essential component required for them. For example, we start with the first level of regulation of gene expression. What do you need to do with the bringing the components? Also understand whether disruption or introduction of them may cause up or down regulation of gene expression. That should be your focal point. So before that, you have to bring in the ingredients. Yes, you have to bring in all the ingredients of what is required for initiation, what is required for post transcriptional processing, what is involved in epigenetic modification and RNA editing. Then you got to understand that what are the function of each components. Unless you don't know the function, you will not know whether disruption or introduction of them will lead to alteration of gene expression. For example, can you tell me quickly what would all be key for initiation of transcription? Remember, we talk about transcription of mRNA because mRNA leads to protein expression. So here you are not doing all transcription process like the MSK1. You're not bringing in all the process and learning on them. No, you are just picking those components and connecting with them, whether they are altering gene expression in a way, how are they upregulating, downregulating, or what is their major function. So who will tell me for initiation, what all comes to your mind are the important essential components, because this is the first level of regulation of gene expression. I just want to see how your focus is. Anything that comes to your mind is essential component. Your cis elements, tata, promoter, enhancers, silencers, their function, RNA polymerase. Yes. Then your transcription factor, absolutely. So you got to bring all of them. One thing I want to clarify, somebody asked me in MSK 2, does transcription need helicase? I would say yes. Remember, transcription factor H has helicase activity too. Remember, unless you know do helicase, they will not unwind. Yes, RNA polymerase is essential. because it moves on the template strand. But what does it all need that is important in regulation of gene expression is to read the cis elements, the tata, the promoter, the enhancers, the silencers to effect regulation of gene expression. So is RNA polymerase to do that has to be activated? Absolutely. Has to recognize promoter? Absolutely. Promoter is to initiate. What is enhancer? To enhance or repress. Promoter is to initiate. What is enhancer? Enhancer is to enhance or repress. Enhance. And does RNA polymerase have to be activated? Absolutely. It's activated at CTD terminal by phosphorylation. So you bring in everything. You bring in the RNA polymerase, the transcription factor, all the cis and the cis elements, where is promoter, enhancers, silencers. This is what I said is transcription factor H. So you know BDFH. H also has helicase activity, and yes, helicase is required for transcription. Now, you're not bringing back these process and drawing them and wasting your time. All you're trying to do is if our initiation regulation of gene expression is controlled at initiation, you need promoter, then you have to see what is the position of promoter, because remember 5 prime affects the N terminal finally, 3 prime affects the C terminal finally. So where is promoter, N or C terminal? Where is promoter, N or C terminal of the sense strand? And sorry, promoter affects N terminal, but where is promoter? 5 prime or 3 prime end of the sense strand? 5 prime or 3 prime end of the sense strand? 5 prime and the end of the sense strands. So you need to understand. Enhancer silencers are at both sides. This is the coding region. Sorry, this is the coding region. Then comes regulation of gene expression. So altering poly-A tail. Okay, let me go step by step. So let's first do initiation only. So for initiation, we'll come to the transcriptional processing, which is the second level. First level is just initiation. So yes, you identify RNA polymerase, it has to be activated, it has to recognize promoter, it has to have enhancers and silencers to do up or down regulation of expression respectively. So you need RNA polymerase. Then the second level of gene expression go to that, which is transcriptional processing. So for transcriptional processing, what all can you modify that will alter gene expression? What all you can modify for the core post-transcriptional processing that can alter gene expression, please? No, I want to know the processes. Processes. What processes can be modified? What all is involved in core post-transcriptional processing? What do you do? Acetylation and deacetylation is epigenetic modification piece. Don't go wrong. There are 4 levels of 5. Yes, there are 4 levels of gene regulation. They are controlled at initiation, post-transcriptional processing, epigenetic modification. When you're saying acetylation, deacetylation, methylation of cytosine, that is not the processing, that is epigenetic modification. Yes, somebody said rightly, adding of tail 3 prime, cap 5 prime, and splicing. Can you alter that and have different protein expression? Yes. So there comes this cap, this tail. So which one is for stability at 3 prime end? What gives stability at 3 prime end? What gives stability at 3 prime end? Poly-A tail. If poly-A tail is mutated, if poly-A tail signal is mutated, stability will be affected, unstable RNA, and that is why if this RNA, mRNA is unstable, what will happen to gene expression, up or down? If mRNA is unstable, down. So, right, you have to pay attention on up and down, but you have to recognize what is the function of these components. Cap is also for protection, but stability is poly-A tail. Is this coding region? Is this coding region between the start and the stop codon? So, do you know your stop codon? Ua, ua, guga. You still need to know them because they come back in your regulation of gene expression. If the coding region is altered, what will be altered? If the coding region is altered, is the length altered or the sequence of amino acid will be altered? If the coding region is altered, what would be it primarily affect? Always have primary effect, the sequence of amino acid. Naturally, if poly-A tail is altered, sequence will not be affected. Poly-A tail is affecting the length. So understanding what are these components, if they disrupt, what is their outcome? Please do not memorize example of troponin, tropomyosin, calcitonin, calcitonin-related peptide. No, you got to understand that it's second and third order. You have to understand the concept, not first order. First order questions will not be tested because we have limited number. What will be tested is second and third order. So please don't do memorization question. Yes, promoter function, enhancer silencer function, then disruption or introduction or removal outcome on regulation of gene expression. Second level was processing. So yes, we did talk about cap, tail, and splicing. You could do alternate splicing, but understand that capping is towards 5 prime, tail is towards 3 prime, promoter 5 prime, enhancer silencers 3 prime as well as 5 prime. Position is important because each of them affect different terminals of your peptide. Does that make sense? So we introduced all of them. We introduced know the function, bring in the essential components. We are still at transcription, and this is exactly what is given in Dr. Binstock's slides. Can you alter the poly-A signal and there could be some diseases? I don't want you to memorize the disease, but I want you to understand that altering poly-A tail signal, mutation in poly-A tail signal, has Dr. Binstock related them to disorders or disease? Yes? Yes or no? Why? Because altering poly-A tail signal will result in what? The outcome you should know will result in what? If you alter the poly-A tail, that will result in unstable mRNA transcript, not changing the sequence. So you got to understand the role of each component, whether it's at the level of transcription, transcriptional processing. Please see, although I'm bringing these diagrams, only one which you need to concentrate on is on mRNA, because mRNA affects protein expression directly. So we did talk about RNA polymerase, bringing all the factors. So yes, at the level of initiation and transcriptional processing, you got to understand regulation of gene expression. Now people who are talking about acetylation, deacetylation, methylation, yes. So now you got to understand epigenetic modification at the level of DNA itself. So methylation of cytosine of DNA will silence what? Methylation of cytosine of DNA will silence what? Does it silence something? Is there anything near the CPG islands? Promoter, because CPG islands are near the promoter, and where is promoter? Towards switch end? 5 prime or 3 prime? Promoter. Promoter is 5 prime end, right? 5 prime correspond to N, C prime to 3. So this is one modification, epigenetic modification. Epigenetic modification means you are not altering the sequence, you are still affecting the DNA. Second is the one which you are talking. So acetylation or deacetylation of histone, which leads to upregulation of gene transcription or regulation of gene expression. Acetylation, acetylation. Acetylation you're doing of DNA or histones? This acetylation, histone. So you have to be mindful. Modification, this is epigenetic modification. Initially, when I asked you what is involved in the post-transcriptional processing, you were saying methylation of cytosine, acetylation, deacetylation. Now they come under epigenetic modification. We talked about them, right? So I'm just bringing your attention. I'm not teaching you and seeing how much you know too. So we did talk about epigenetic modification. You got to understand outcome. Is there silencing downregulation, or there is which is by methylation of cytosine, or is it acetylation of histone? Upregulation, you got to understand that. Should you bring all your trans elements? Absolutely. Their position? Yes. Their function? Absolutely. You got to understand their function. Do you know all the trans factor? So which trans factor binds to tata? Which trans factor binds to tata? Trans factor D. So does trans factor help in any initiation of transcription or transcription? Absolutely. Your promoter, RNA polymerase has to recognize promoter, and hence the silencer have to work, trans factor have to work. You should know all the cis versus trans elements. We identified that. Yes, anyone? Yes, transcription factor D binds to TARA. So you have this complex of transcription factor. This is what I ask you. See, transcription factor D binds to. Sorry, just let me get my pointer, laser pointer here to help you understand that. This is what I was asking. Transcription factor D binds to TATA. Do you need RNA polymerase? Absolutely. Do you need the transcription complex? Yes. Enhancers, they bend. They are distal, very distal. They bend and interact with the DNA. Enhancers bend and interact with the DNA. That is their quality. So they all have their function, their specific quality. We are not talking about final, we are talking about primary effect. If you're altering poly-A tail, the resultant primary effect is, please don't go to the second, third, fourth, fifth effect. Know the primary effect first. So when your question is what is happening with altering poly-A tail, length can be altered, but first of all, it is altering the stability. Primary is stability is affected. If stability is affected, what will happen to gene expression? Up or down regulated? It's not altering sequence, please. Sequence is altered when the coding sequences are altered. That alters amino acid sequence. Poly-A tail is for stability or length. Did I clear that, please? Yes, poly-A tail terminates, so then it will be a different length. Diego, did I clear that, please? No, it's not altering sequence. Yes, focus on the primary effect. This is very good question, though, somebody has brought up. Primary enzyme for the disease, primary mutation, primary effect. Please, more and more you focus on the primary outcome, better grades. For example, poly-A tail, unstable mRNA transcript. Then, if asked, go further, because as you go further, everything becomes vague. What is concrete is primary outcome. Does that make sense? Yes. After that, it could be variety of things. So we don't want to open that Pandora box. Does that make sense? I want you to become aware, more and more focused, please. No poly-A tail, you are not altering sequence, unstable or length. Okay, so again, we were coming to this. So you should know, see, enhancer is different. It bends and interact with DNA. It enhances the function of outcome of regulation of gene expression. So you got to bring all the trans factors, H-head helicase activity, as I told you, you all recognize that transcription factor D binds to TATA. What about coactivators? So you bring in everything. So does coactivator bind to DNA? Does coactivator? No. So what is the function of coactivator? To keep the transcription factor together. So does coactivator help in initiation and transcription? Absolutely. A coactivator disrupted may disrupt transcription, yes. So unless you bring in the ingredients and know their function, you cannot order third order, which is outcome of if affecting them and gene expression. So you should know coactivator function, what are they doing, what are enhancer doing, how they are helping in transcription. Then only you will understand trans factors, BDFH, their function, how they are helping. Then only you will understand their outcome if they are disrupted or introduced. For example, introduction of a silencer will upregulate or downregulate in the exon in introduction of a silencer. in an exon will upregulate or downregulate gene expression? Downregulate. So that's a second order outcome. But before that, you got to bring in all the ingredients. We have done all trans factor, BDFH. Yes. Coactivators, they do not bind to DNA, please. They bring in the transcription factor together and help the transcription factor complex to complete transcription initiation. Look at enhancers. They bend the DNA because they are very distal, so their positions are important. You see here transcription factor D binds TATA. You see RNA polymerase activated. Here you see coactivator, which does not bind to DNA. So this is a very good diagram to tell you what is the different components, what are cis elements, what are trans factor, what are coactivators. RNA polymerase is essential. So why are we talking only about RNA polymerase 2? Why are we talking only about RNA polymerase 2? B is the bridge between D and F. B is a bridge between D and F. D and F. Somebody's asking me what's the function of B. Yes. D and F, please, because D binds to TATA. Now, you can say B is a bridge between D and TATA. D is binding to TATA, right? B is a bridge between two transcription factors, D and F. D binds to TATA. RNA polymerase activation is essential. We talked about that. You know all of them now. TATA, enhancers, silencers, promoters. What are they called as now? If I ask you, what are they called as? TATA, enhancers, silencers, promoters. What are they called as? All of them together are cis elements, and there are trans factors too. Bring the trans factors, we recognize them, enhancers, bend the DNA to interact. Now, motives. Why do you need motives? First of all, do trans factors bind to DNA? Do all trans factors bind to DNA? Or there's only some, no. Which trans factor binds to DNA? Transfactor D. But do trans factor interact with DNA? Do they interact with DNA? Can trans factor interact with DNA? Yes, trans factor interact with DNA. So for trans factor to interact with DNA, they need motifs. Like clamps, they need motifs. So you need to recognize those motifs. Without them, trans factor cannot interact with DNA. So these are the trans factor, glucocorticoid receptor, SREBP. They are all trans factors and they need respective motifs to bind to DNA. So we have discussed motive. So why should trans factor D need motifs? Because trans factor T binds to DNA, right? Motifs are required for some trans factors who want to interact with DNA. Specific only trans factor needs motifs, right? Like steroid hormone receptor, if they want to interact with DNA. For example, SREBP, if they want to interact with DNA, they need motifs. Does that make sense? There's difference between binding and interacting. Transcription factor D binds TATA. Interacting means they want to act on their responsive element and affect transcription. Did I clear that? Great. So if some trans factors want to interact with DNA to affect a regulation of gene expression, they need motifs. We talked about these motifs. Here, steroid hormone receptor is acting as a trans factor to interact with the DNA. Sorry. And this is steroid hormone receptor acting as a trans factor to interact with DNA. Here is a steroid hormone receptor acting as a trans factor to interact with DNA. And see, you don't interact with the whole DNA. That's why it's known as hormone response. You interact only with a segment of DNA, known as hormone responsive element of DNA. So we did talk about motives. Please don't worry about the intracellular cholesterol control. In the previous curriculum, biochemistry was taught before genetics. So students knew everything around this slide. But right now, concentrate on, do you need some proteins? I would call them proteins. Some motifs. If the proteins want to interact with the DNA, do they need some motifs? And SREBP is also one of the protein. And we are not asking you to memorize regulation of cholesterol controlled by SREBP here. So understand that. Here again, see, we were talking about, so you are trying to bring in the components and look at this. This is down regulation. So I'll just ask you, does phosphorylation of RNA polymerase cause down regulation? Does phosphorylation of RNA? No, it will not cause downregulation. So you got to understand the outcome. And when you bring in the components, you will understand why deacetylation of histone will cause downregulation. So you have to bring in the components and then assess the outcome. So please, though, that baking of the cake is required, whether at the level of initiation, transcriptional processing, the epigenetic modification, and RNA editing. Bring in the components. Just a second. Okay, let's do this question. Can you answer? I'm not sure. Can anybody unmute and tell me you can answer? Yes or no?
I think so. If you go on the poll
Yeah, we
over there.
can.
Great. So let's see this and then discuss it. So understanding how we can bring in all these components together, understand their function and then outcome. There's always one best answer. There might be two close answer, but as you become more focused and repeat your focus, that distinction goes low and low. You got to pick the best answer. Everybody is good?
To enter it manually.
Oh, no worries, no worries. Do it. I just want you to understand, like, I think students wanted few questions, right? Just wanted to see where you are going with these lectures.
But yes, we do want more questions actually.
I'm trying. There's a lot of help. Biochemistry genetics is giving a lot of help, huh? All you need to is grab it. So let's see. Everybody's good? Anyway, this is not for grading, please. I will see your attendance by your participation. Okay? Is everybody good now? So we can finish the review. You want me to discuss it, everybody?
Yes, please.
So see, we are trying to understand how do you repress expression. So would you choose enhancers or silencers? Because these are the two close. Am I right? What do you want to pick, enhancers or silencer? Yes. So the correct response is, let me see the response rate. If I can hear, can you see the correct answer? I think this is my first time doing it via. Can you see the correct answer? No?
No, we can't see it on your side.
Okay, great. Maybe I need to work a little more on, but the correct answer is silencer, right? Everybody pick silencer. Too close would be silencer or enhancer, but the correct answer should be silencer. Everybody is good with that? Oh, wait, I can see that now. Okay, so 94% chose silencer. I'm good. Are you seeing the response now? Yes?
We see that it's closed, but we don't see like the correct answer.
Okay, maybe that's only at my end. So I'm glad it's 94% silencer, and the correct answer is silencer element. Does that make sense, right? Are you understanding where we are going towards identifying each component and their outcome? Yes? Let's go further. So yes, silencer should be the correct answer. Within the exon is very important because they are all cis elements, Joy. We are talking about the cis elements here, whether it's promoter, enhancers, silencers, right? We are not talking about them as a trans factor. Yes? Great, great. So, yes, can I take up the questions later, please?
Okay.
Yeah, I'll hold on the questions so I can finish the review and do some questions. So altering poly A tail, remember we talk about poly A tail 3' side affecting C terminal. You can alter the length. There are examples, though I don't want you to memorize example. When you alter tail, alter splicing, or you alter both. But splicing can change length and have different exons that you need to incorporate and different protein expression, uh, which is also an example of how you can alter them, have a normal outcome and abnormal outcome. Again, 3' affecting stability. 5', yes, what is that 5' end? Promoter. Where is promoter? Promoter is where? 3' or 5', affecting expression, affecting N terminal. Then you have this coding sequence, which is actually alteration may lead to alter protein sequence, structure, and its function. Please keep your question. If it's quick, I will definitely answer them here. Right? Doesn't make sense. So identifying what are the components, what is their where are they on 5', 3'? Are they affecting NC terminals? Are they affecting length? Are they altering the sequence? That is how they will alter the expression, gene expression. Does that make sense? Right? For example, there are the example of protein that can be there. And you can form protein by altering either splicing. There are examples altering promoter, altering splicing and poly A tail. I don't want you to recognize or memorize those examples. For example, it's given here alternate splicing, troponin, tropomyosin. Alternate promoter. Have you learned any protein that you form by altering a promoter? Have you learned any protein that you form by alternating promoters? Any protein expression? Anyone? Yeah, dystrophin, and dystrophin is a dystrophin gene is the largest gene subjected to a lot of mutation, and altering N-terminal results in dystrophin tissue expression. So there are example, but all the main component is what are poly A tail required for? They're at what site? What will they affect? Can they affect and you will have disease? Promoter, what side? What is the function of enhancers, silencers? Rather than memorizing examples, no, this is what I was asking, remember? Dystrophin, different tissue expression is by altering the promoter and N-terminal. You have a CV of dystrophy. There are Duchenne's and Becker's. You have Duchenne's muscular dystrophy, largest gene subjected to deletion. Please know that most common mutation. We also did last in MSK1 treatment of Duchenne's patient by using by masking the exon, converting Duchenne's into Becker. Becker is better, nothing is better, but Duchenne patients can be treated by the antisense oligonucleotide masking the exon. The last one is, so just to summarize, we have regulation of gene expression at the level of initiation, transcriptional processing, epigenetic modification, and last is RNA editing. Single gene, remember, two product. How can you form a ApoB100, which is 100% translated, versus ApoB48, which is only 48% translated? This is because of RNA editing. Please know RNA editing comes after RNA is being synthesized by RNA polymerase 2, pushed out into the cytosol. RNA editing is happening in the cytosol. So this is all about regulation of gene expression. This is the summary. You got to understand the cis elements, their function, their position, 5 prime corresponding to an N terminal and 3 prime corresponding to C in protein, right? I hope everybody is good with regulation of gene expression. This was the largest segment. Let's quickly do now fibrous protein and then glycoprotein and proteoglycan. I hope this is helping. This is how I thought I would review, just recap concepts. Is that helping everyone? Is it good? Okay, great. Take your help. Help is everywhere. Let's do this.
Professor, will you share this set of slides as well?
Yeah, I think I've shared all my recap slides actually, but I may reshare this one. I've shared all on the drive. These are the same ones I went through, but I'll reshare them again with
It's
the.
on Canvas now, this whole thing.
Sorry?
It's on Canvas.
No, this is on the help folder with your class reps. This is not on Canvas, please.
I just downloaded it from Canvas, Dr. Bach.
Okay. I'm glad if it's there. Maybe they put it on Canvas. Okay, let's see that. I'll clarify that. But I thought that this is with so I put two files. One I've put for the extra help, which is on your help folder on Drive. Second, I put on the Canvas. But I'm not sure if this is also on Canvas. No worries, but it's okay. That's good though. Stick to your learning objectives. Fibrous protein, what did we talk about? Fibers of fibrous protein. First of all, bake the cake. Identify the collagen processing steps. Steps that are extracellular. Then bring in the nutrients, vitamin C and copper. You got to bring in your nutrients to have a good cake. Then I sit with EDS, osteogenesis imperfecta, alpha-1 antitrypsin, and am I missing something? I'm missing one. Menkes kinky hair disease. That's the icing. So we recognized what collagen is made up of, every third residue. Glycine, remember, simplest, smallest. And replacement of glycine by bulkier amino acid, can it result to disease? What was the disease? Where replacement of glycine led to a bulkier amino acid. Replacement of glycine led to bulkier amino acid. A replacement of glycine by a bulkier amino acid resulted in disease, osteogenesis imperfecta. So then do a differential between osteogenesis imperfecta and EDS. Also do a differential between osteogenesis imperfecta 1 and 2. Very good. You're very specific and very focused that it's osteogenesis imperfecta 2, not 1, where there is qualitative defect. Glycine replaced by bulkier amino acid. In 1, it's quantitative, where the chains are low, alpha chains are low. EDS, the enzyme. EDS, yes, EDS is mainly because of the collagen processing enzyme defect. Absolutely. So we identify glycine as the simplest, smallest, most common amino acid, fits in. If replaced by bulkier, unstable, alpha-1 more than alpha-2 double. We identify the processes, role of vitamin C versus copper. Both are required for strong collagen. Up to triple helix formation is intracellular, then it is secreted and then becomes extracellular events. Please, we also compared collagen versus elastin. There was only one difference. So what does elastin has, collagen does not? This is what we do in our life all the time, right? What I have, what others have, I don't. So what does elastin have, collagen does not? What does elastin have? What is in elastin that's not in collagen? Anyone? What is an elastin that's not in collagen? Anything is unique to elastin which is not present in collagen? Did I lose you? Desmosine, absolutely. Desmosine is responsible for stretchy property of elastin to stretch and come back, which is not seen in collagen. Collagen is the aldol crosslinks. So then you understand why is it different? Why are the enzymes? So do you talk about peptidases? Do you talk about peptidases in collagen processing? Do we talk about peptidases in collagen processing? Yes, so bring in all the enzymes. First bake the cake, collagen processing steps, intracellular, extracellular, peptidases, hydroxylases, glycosylases, and then lysyl oxidase, copper, vitamin C. We introduced all that. I think there is one clarification that do triple helix form first or disulfide peptide bonds first? The step to form the step after hydroxylation. First step is N-terminal hydrophobic, then hydroxylation, then glycosylation. The next step is to form triple helix. So triple helix, yes, is formed first. And how is it stabilized is by the terminal disulfide peptides. So after triple helix formation, disulfide peptides stabilize them. So that is the answer. Somebody asked me a question, I also bring in clarification. You know vitamin C role, you know the processing, you know copper, you know the difference between collagen and elastin. I'm not teaching you, bringing your attention, right? Collagen is degraded by collagenase, elastin is degraded by collagen is degraded by collagenase and elastin is degraded by elastase. So you got to know elastase too, collagenase, lysyl oxidase, hydroxylase. Sorry, let's do this one. I will just give you a minute and please, I will then explain you. Okay, if you can answer, you answer. Otherwise, you can listen to the discussion. Is that fair? Everybody? Okay, so we are understanding that somebody asked me, is triple helix form first? Yeah, triple helix is formed but stabilized by the terminal disulfide peptide. So let's see what is essential for that step. Forming triple helix, what's essential for that step? I will give you a minute, then I will answer because I don't want to waste time. I think we are taking too much time in analyzing whether you can get into pole everywhere or not. Because I want to finish glycoprotein proteoglycan. I hope you're following my pace and this is working. Please let me know. Okay, guys, let's see that, right? First of all, let's see, we are talking about triple helix. So should I talk about ascorbic? Can you unmute and answer because I can see your chats and the screen at the same time. So tell me, for triple helix formation, do you need ascorbic acid, which is A? Yes or no?
No.
No. Do you need alpha-1 antitrypsin?
No.
No.
No. Do you need RNA polymerase 2
Yes.
directly to assemble them?
No, that's
To
for
assemble triple helix, do you need the RNA polymerase 2? Yes or no?
No.
no.
No. Do you need RNA editing? An ApoB gene? What are the chains here? Alpha chains, right? So is RNA editing ApoB gene correct?
No.
No.
So that is why what is your correct answer is this step requires formation of terminal disulfide peptide, which starts at C terminal and finishes at N terminal. Do you need disulfide bond for it?
Yes.
Yes, so that should be your correct answer. So you understand that there are processes that require specific buzzwords are there. You can't talk RNA editing and collagen processing, triple helical formation here. Does that make sense? So what is your correct answer? Let me see those who are, I hope it's 90%. I'll tell you what's the 88%, which is not bad. Okay, so your correct answer is C and N terminal peptide and disulfide bond. Does that help you? You have to be very specific, right? I can take up questions later on. So we did talk about Menkes, kinky hair disease, and copper distribution defect. Did we distinguish osteogenesis imperfecta from EDS, which is brittle bone, trivial fracture, quantitative. That is osteogenesis blue sclera. That is also osteogenesis imperfecta. We identified them as collagenopathies. We did that. Did we talk about desmosin, which is only in elastin? Yes. But desmosin also need the same enzyme lysyl oxidase and copper, but desmosin is not in collagen, right? Desmosin crosslinks are in elastin. Please, many students are confused between Marfan's and EDS. There's no correlation at all. Marfan's is slender body physique, long arms, dislocation of lens, which is upwards. They also have large aorta involvement. So if Marfan syndrome is is a defect in collagen or elastin, Marfan syndrome. Marfan syndrome, what do you relate it to? Collagen or elastin? Elastin. And which protein is defective? Fibrillin. So we talked about all of them, right? Elastin is degraded by elastase. Please ice the cake too, with all disease, all the disorders that have been addressed in the lecture, right? Hope this is helping. Last is your glycoprotein, proteoglycan, and that finishes your recap, your review today, okay? Let's do glycoprotein and proteoglycan quickly. Do you need a break or should I finish this? It's just maybe 10 minutes. Is that
No
good?
break so that we can, because we might have other stuff ahead.
Great. So let's do it 10 minutes, okay? Let's identify glycoprotein. I hope this is helping. I just wanted to refresh everything. Everybody's good? Okay, let's finish. Let's do glycoprotein versus proteoglycan. First of all, please stick to your learning objective. Let me tell you, key of glycoprotein, proteoglycan, you got to contrast glycoprotein, proteoglycan with structure, function, synthesis. First, you contrast glycoprotein, proteoglycan, then you can contrast O versus N-linked glycoprotein. Compare and contrast is important. Then you what I sit with the disease. Your hurlers, hunters, eye cell disease. Does that make sense? Yes. Also, some terms that you should know. So let me quickly finish glycoprotein versus proteoglycan. Any question here? I'll open for question and answers. First of all, identify, you know the sugar. So I'll just ask you, what's the sugar in proteoglycan? What's the sugar in proteoglycan? GAGs. What's the older terminology for GAGs? GAGs, MPS. So do you know all mucopolysaccharidosis? Give me some mucopolysaccharidosis. Give me some mucopolysaccharidosis. Hurlers, hunters. Absolutely. Excellent. So you just need to be focused. What is my sugar in proteoglycan versus glycoprotein? Are they negatively charged and that's responsible for function? Absolutely. Do you know GAGs and their function? Do that. Physiological or clinical. You got to understand that they are synthesized in the same order and degraded in the lysosome. What is added last is removed first. You need to understand GAGs and MPS are part of proteoglycan, not glycoprotein. Yes, you don't need to memorize the core, but you have done aniline glycoprotein synthesis. So what's the core in aniline glycoprotein synthesis? Is it xylose, galactose, galactose, or pentasaccharide core? What's the core in aniline glycoprotein synthesis? Is it xylose, galactose, galactose, or pentasaccharide core? pentasaccharide core, right? So understand the difference between O or N-linked glycoprotein synthesis. Dr. Panini has talked about pentasaccharide core there. You know the function. Please know the function of GAGs. GAGs are proteoglycans. O and N-linked glycoprotein examples are good, but please don't miss high mannose, which are example of lysosomal enzymes. What is the disorder associated with lysosomal targeting defect with lysosomal enzyme? I cell disease. So definitely you should know this example. Did I tell you where is pentasaccharide core? You have learned in N-linked glycoprotein synthesis. Pentasaccharide core is with N-linked, not with O-link, not with proteoglycan. So some specific terms like GAGs, MPS, their function, where is pentasaccharide core, difference between O versus N-linked glycoprotein synthesis before you go to Hurler-Hunter's or eye cell disease. You know cell's function, glycoprotein, extracellular matrix, proteoglycans. So why are they, where is sugar negatively charged? That is responsible for shock absorbent. Give me a sugar which is negatively charged, attracts water, and is a shock absorbent. Hyaluronic acid. So hyaluronic acid is in proteoglycan or glycoprotein? Hyaluronic acid is in proteoglycan or glycoprotein? Is a GAG. So it's in proteoglycan, right? So similarly, identify other GAGs. I will get to that slides. So shock absorbent, negatively charged GAGs is hyaluronic acid. Have you done all these GAGs? Absolutely do them. Chondroitin cartilage, keratin cornea, dermatin skin. This I asked you. So you got to understand the function and role of these GAGs. GAGs is a sugar in proteoglycan, right? I've enumerated some GAGs too, whose function you should know. Bottle brush, proteoglycan. GAGs, proteoglycan. This is also one of the function of heparin. Heparin is also GAG. Heparin is also GAG. Degraded lysosomes. If you can't degrade them, lysosomal storage disease. Did we compare mucopolysaccharidosis with eye cell? Tell me what is seen in eye cell, which is distinct than mucopolysaccharidosis. What is the difference? Eye cell disease finding. What is the different eye cell disease finding which you will not see in single enzyme deficiencies like Hurler's and Hunter mucopolysaccharidosis? Lysosomal enzymes everywhere. Aggregation in the lysosome is very important because eye cell disease does not involve single enzyme. It involves many lysosomal enzymes which are not targeted. That is why the debris that accumulates in lysosome is not single substrate. It is mixture of GAGs and glycolipids. That is why it's known as inclusion bodies. They are known as inclusion bodies. Yes, lysosome's empty, lysosome filled with debris because enzyme are missing, lysosomal enzymes everywhere. It's not a single enzyme deficiency disorder. It is a lysosomal targeting defect, right? So there's a difference. You have done Hurlers and Hunters who have a good eye and they hunt for an X. Who have a good eye and hunt for an X. Hunters, is it a single enzyme deficiency disorder? Yes. Which are the GAGs that contain iduronic acid that accumulate in them? Which are the GAGs that contain iduronic acid? Heparin and dermatin sulfate that accumulate in them. So you have done Hurlers as well as Hunters. Then distinguish function, glycoprotein, cell surface, recognition of cell and bacteria, blood group antigen, they're all glycoprotein. Has Dr. Panini distinguished O and N-linked glycoprotein synthesis? Absolutely. So who has post-translation modification, O or N-linked? Post-translation modification. O-link. So he has compared and contrast O versus N-linked glycoprotein. I have also explained you N-linked glycoprotein. This is the pentasaccharide code that I was talking about. I have also explained you N-linked glycoprotein by this slide. So for N-linked glycoprotein, what are components you need? Yes, you got to compare and contrast it with O-linked glycoprotein. Post-translation is O-linked. Co-translation is N-linked. Pruning starts in the ER, completes in the Golgi. So should you compare and contrast? Absolutely. See the components required for O versus N-linked glycoprotein synthesis. And what is lysosomal targeting defect? Are you clear with that? Lysosomal targeting defect is eye cell disease. Have we done eye cell disease? Yes. Please understand the disease. Yes, presentation is very important for you to know, but you should go back to the defect. Defect should be clear in your mind. If you have any question, I can explain you. So eye cell disease is a lysosomal targeting defect. Yes, whereas the signaling to target lysosomes to their destination is missing. There's the deficiency or defect in adding a signal. You know there's always a signal. When these proteins are synthesized, they need a signal to go to their destination. Without signal, they will not go to their destination. We discussed that in MSK 1, 2. Eye cell disease, look at this. I was talking about this inclusion bodies. Please see there is not accumulation of single substrate here. There's a lot of debris. That is why it's known as eye cell disease. Empty lysosome, lysosomal enzymes everywhere. Let's do this one. We are talking about hurlers, so we will do all the differential. I'll give you a minute and I can discuss this. This is my last question. And then I can open the forum for question and answers. I hope you followed my pace. And I will give you a open forum for question and answer too. I can answer them. I can reshare these slides. Okay, let's see this. This is Hurler syndrome, okay? So let's go over each choices. Kinky hair, is it Hurler syndrome? Please, you can unmute and answer. Coarse kinky hair, is it Hurler's?
No.
Brittle bone, is it Hurler's?
Osteogenesis imperfecta.
Osteogenesis imperfecta. Are we thinking about smoking history here in Hurler's?
Emphysema.
Emphysema, we are not thinking here. Now, so what are you caught between in only these two? Because both have coarse facial features. Which one should be correct? A or B?
A.
A, because apart from coarse facial features, hurlers have corneal clouding. Large inclusion body would be which one?
Eye cell disease.
Eye cell disease. Does that answer your question? Right, you have to understand. Very good. 96% answered coarse facial feature and corneal clouding, but it's not that you just recognize clinical signs and symptom. Please also do the defect. Okay, sorry. That is the correct answer, which is A. I'm glad 96% answered. I wish you good luck. I hope this helped.
So I have a quick question, if you mind.
Yeah,
though.
yeah, sure. Yeah, now you can
So,
ask.
it's kind of like tagging off what Miss Joy asked. I understood that it was a silencer element when the cis bind. I was just thinking of another way to like twist the question. If a cis or trans element binds to an intron element, because of
I think it should be exon.
no, I
because
know,
the-
but hold on real quick. Because of splicing, nothing would happen, correct? Because the introns are spliced out. You get what I'm saying? Like,
Yes,
sorry,
absolutely.
I'm trying to combine two concepts in one in my head. Like,
Sure. But don't overthink, right? Like that on the
okay.
test.
I was just wondering, I was like, okay, what if it said O instead of
On the
T?
test, read it as it is. They're
Okay.
talking about a silencer on the exon. What is my function of silencer? What would be the outcome?
Okay.
How will silencer repress? Rather than saying, okay, what if it was an intron? What if you get my point? I understand how you're doing it now, but not on the test, right?
Okay, perfect. No, I'm just trying to, because y'all say make up multiple questions or one question. I'm like, okay, how can I flip this? But thank you so much.
I never encourage people making multiple questions from one question. I always encourage students learn 5 concepts from a single question. I think that's actually very dangerous to make multiple question from one question because you might not frame it,
Okay.
might not. So I don't encourage people. I encourage to learn 5 concepts from a question. I'm just clarifying what I
No, I
encourage.
understand. So
Yes.
if that part by accident was spliced out, then nothing would happen, correct?
If the axon was spliced out,
By accident, you know what I mean? Like,
or if they ask, but I,
yeah,
you know,
yeah,
before,
they say like, oh, due to because there was a question similar to that on Canvas that you provided with us, where it was like, okay, now the splice sequence was 1, 2, 4, and 6 versus 1, 4, and 6 because it spliced it differently. Um,
here the splicing is not applicable, right? So I don't want to confuse you like that. You get my
okay.
point?
No,
There's
perfect. No, that's totally fine. If I don't need to go there in my head, I won't.
yes, 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 yes.
Thank you.
Okay, tell me yes. Other questions? Anyone can pitch in. Yeah, tell me please. Joy.
Doc- Hi, Dr. Ba. I just have so when we talk about osteogenesis imperfecta,
Yes.
type 2 and
2.
type 1, you say type 2 is qualitative versus type 1 is quantitative defects. What do you mean when you say qualitative?
By that I mean, usually most of the collagen processing defects are qualitative. That means, and most common is glycine replaced by bulkier, which is seen in osteogenesis imperfecta 2. There are very few example of quantitative defect leading to collagen disorders. One of them is osteogenesis imperfecta 1, because collagen is made up of alpha 1 and alpha 2 chains. Alpha 1 is double of alpha 2. So if they are affected, if their quantity is low, it disrupts collagen synthesis. That is why that's what I mean by quantitative. That means chains are not even synthesized. Others are qualitative. That means after the chain is synthesized, they are not being modified. Does that clear your?
Yes, that actually helped a lot.
Yes.
And
Quantity means they are not being synthesized only. That's called quantitative defect.
oh, so change. Okay, thank you so much.
So collagen is triple helical structure but has 2 chains, alpha 1, alpha 2. Yes, Joy?
Hi. So okay, so that question, I I think what confused me was like because it said within the exon, it was asking about the cis element within the exon, and I think that's what threw me off because
So what
well, I'm
would
just
you
wondering.
choose? What would
Well,
you choose? That enhancer within the axon. So if the enhancer is within the axon, its function changes according to you?
no, but no. So like what threw me off was the within the exon, and that's what I'm wondering about. I thought cis and I thought whether it's I thought cis elements were still outside of the gene area.
Yes, yes, cis are part of DNA, but this is an experiment. Theoretical question. It's just asking you that this cis, which is also part of exon, how will it repress the function, right? I think you might be overthinking in localization, right? But the
Yeah.
question was asking the function.
Okay.
How does enhancer act? How do silencers act?
Yeah.
Right?
Yeah. Okay. Thank
But
you.
but I understand that. I think you might think that okay, if there was other position, what would have been the effect? But no, this is simply
Yeah.
primarily asking how does silencers act and how do enhancers act? They don't act in the same way. Remember, enhancers bend the DNA.
Yeah. Yeah. I mean, they're like, but the cis elements, they're still outside of the general gene
Yeah,
region.
but this is an experiment asking you if this was, right? For example,
Okay.
if and but, right?
Yeah.
Could be
Okay.
anything.
Yep. Yep.
Yes.
Thank
Diego?
you.
No worries. Diego?
Jennifer was ahead of